Pleasure to introduce our next speaker, Dr. Wendell Lim, Professor and Chair of Department of Cellular and Molecular Pharmacology at University of California, San Francisco. Wendell.
Okay. Thank you very much. Thanks to Kathy and to everyone Mike for organizing this really interesting meeting. I apologize that I wasn't here yesterday. It sounds like there were really interesting things, and perhaps I'll touch on things that are related to I think, sure I will, some of what was discussed yesterday and today. So it's great to follow Veronica because I'm going to give a very conceptual talk, also multi-scale, multi-system, et cetera. What we've been interested in is using approaches in synthetic biology to really try to more maybe proactively and empirically interrogate kind of the design principles of complex biological circuits. And so I'll talk about our efforts today to try to apply this to questions in development or morphology. That is, how is it that we can try to actually forward program at a genetic and molecular scale things that then can determine higher order tissue formation, multicellular tissue formation, in order to try to explore some of the principles that underlie, or common principles that underlie, multicellular self-organization. Okay, so I think we're all interested in this problem because, of course, biological systems have this amazing way of going from a single cell that has this genetic program in it and then to become big structures like an elephant. And really, the thing that makes biological systems so different from, say, man-made objects of any type is that they aren't organized in the same way of like taking building blocks and then linking them together in Cartesian space. But rather that they are self-organizing systems that are built up at this multi-scale level, as we heard from Veronica. And so one of the things that we're interested in is the idea that are there underlying all of these different kinds of morphologies we see in different animals, different species, I'm sorry, that there are there sort of basic primitive operations, one like intrinsic cell polarity is one of those things. And can we try to understand those and understand how, of course, how they're deployed by deconstructing existing morphologies, but really we'd like to also not just take apart things, but ask how do we actually take those rules and ask if we can understand them well enough to forward, maybe evolve or program new kinds of structures. And the idea here, again, is that there are these sort of molecular words, and maybe there are these sort of sentences and grammar and paragraphs, but how do we actually not just deconstruct some great work of literature, but rather start asking how do we write our own sentences and paragraphs? Okay, and this fits into a bigger scheme of how we think about synthetic biology, and in this case of synthetic developmental pathways. That is, traditionally we think of biology as studying what exists out there, and then in sort of the natural history kind of background, trying to understand how they work. But we have been more interested in asking also with molecular components, genetically encoded components, as kind of the substrate for evolution, what is actually possible? What are things that can exist that can do certain functions, and what are things that can exist? And trying to reframe the problem in a more general way. And one of the things that we think is important is that really, if we want to understand not just sort of what exists because of certain evolutionary history as well as those principles, they're often sort of interleaved in a way that's difficult to convolute, that if we can actually start exploring space free from that evolutionary history, we can actually ask more about the physical principles, and these things that can do a certain function must obey the same sort of physical laws, so maybe we can get a cleaner understanding of that, of those kinds of principles. And then also, when we want to have cells and tissues and other things that do useful things, we actually often are fighting against the biology, and we want to do things that are different, say like in the case of regeneration in a context of injury or disease. And so I would argue that if we can understand this space, also there's probably a lot of useful things there that we could use and deploy biology in new ways. So with respect to development, of course, if you take classical sort of developmental systems, much of the work has gone into analyzing how these evolved systems work through a combination of genetics and biochemistry and et cetera, cell biology. There's been obviously a lot of work in the more recent years of trying to use theory to understand and enumerate what are kind of developmental schemes in a more principled and general sort of way. And I think what we want to try to add to that is really tie that together by bringing empirical systems that we can actually start trying to build and compare. And I think that allows us to ask many different kinds of questions that enhance what we're understanding from the other sorts of approaches. We can start trying to ask questions about how different ways that multicellularity could arise, because that's basically what we're trying to mimic in many different ways. Questions about sufficiency. Really, you see a lot of systems, and we can see principles in them, but oftentimes there are multiple mechanisms that are overlaid. Can we start asking questions about what's sufficient to achieve certain behavior, as well as the whole plausibility of how things like multicellular organisms could have evolved? Again, this is cleaner from a structure function analysis, because we're trying to uncouple things relatively free from evolutionary history. And then, as I said before, this may, in this context, give us the ability to ask, how do we actually regenerate tissues, for example, in the absence of the normal developmental cues? Can we actually use other cues or other things like that and write modified programs? And can we also maybe even make tissues that are better or better suited for particular needs? Okay, so typically in synthetic biology, we're thinking about how we program new cellular functions from molecular encoded components. And first of all, we have to think about how is it that we wire individual cells so they sense certain things, use that information to make decisions, and then deploy various things in terms of transcription, cell shape, secretion, et cetera. In the case of trying to start thinking about how we do multicellular things, we of course want to start trying to ask, how do we wire the cells together so they actually start talking in a coherent way? And then, of course, very importantly, how do we actually start taking those genetically encoded molecular sort of components and words, and then putting them together into programs that actually now helps to, in theory, determine macroscopic structure. So one of the things that has really helped us in trying to get into this is the development of kind of orthogonal, very controlled tools for cell-cell communication, as well as cell-cell adhesion and other things that we think are part of the fundamental toolkit for how cells, multicellular systems organize. And one of them is this synthetic notch system, which is based on the native notch juxtacrine signaling system this was developed in my lab by two outstanding former postdocs, Leo Morsoot and Cole Roybal. He's at USC now. Cole is a colleague at UCSF. So we're looking for a new kind of way to put in whatever new connections between cells that we would like. And so Notch, as many of you know, is involved in cell-cell communication, where the native Notch molecule will bind for a ligand like delta on the opposing cell. There's sort of a mechanical pulling that leads to a cleavage of the protein, and then the intracellular part can go into the nucleus and regulate transcription. What we showed is that we're actually able, if we do this correctly, to put on different recognition domains on the outside that, for example, antibodies, single-chain antibodies that can recognize a target that we want. Could be an interesting molecule on a tumor, could be GFP, et cetera, but then put in synthetic transcription factors on the inside. The system still transduces that, but now we can drive off a responsive promoter whatever genetic program we want. So we can have the flexibility to detect something on a neighboring cell in the environment and then turn it into any genetic output, genetically encodable output. We can put different heads on this and detect different sorts of things. We can put different synthetic transcription factors in here, and as long as we have different driving promoters, we can get different input-output responses. And we can actually use these in the same cell because they don't have any signaling intermediates, so that these are actually orthogonal and don't crosstalk with the native Notch or with one another. And we're still limited by what genetic payload we can get in. Okay, so this is just an example that we can take something. This is a tumor antigen involved in B-cell cancers, but we can put a single-chain antibody that recognizes this, put in the synNotch receptor, drive something like GFP, and this just shows this is what it's like, this cell, this receiver cell off, but then GFP can turn on when we present it with cells that present that antigen. We've shown that this works in many cells, from fibroblasts to immune cells. They work in neurons and also in stem cells. So very universal and highly modular. We can put many different things on the outside. So again, we think of this as one of a type of set of tools that we're trying to develop that provide a universal channel for programming in new cell-cell instructions. And it could be using something like Surface GFP as a kind of novel orthogonal channel, or it could be that we read something that exists on another cell naturally, and we try to turn that into a signal that the receiver cell can use. So we've been using these sorts of synNotch circuits to reprogram and explore many kinds of multicellular or cell-cell interactions that we really think really this is one of the big frontiers. Is we kind of understand cells relatively well. I know we don't understand everything, but really I think so much of biology is about the way that multiple cells, of course, interact. And I think everyone here probably believes that. But we've been using it mostly in the context of immunology, of trying to use these sorts of things to explore how immune cells can interrogate different target tissues with multiple sorts of sensors. And that's turned out to be extremely useful in developing new kinds of therapeutic immune cells. We're very interested in sort of the idea of using these things to rewire and modulate, say, connections between neurons, which is a great example of where morphology and the multicellular circuits really determine function, but then also interested in this question of development and regulation and regeneration. And we kind of view this from the cellular perspectives. These are not all that different. They all involve kind of communication and self-organization, but they still use the same sorts of tools. So Satoshi Toda is another postdoc who wanted to ask whether we could write simple programs using these sorts of tools. And when we step back, of course, what are the common abstract features of almost all developmental pathways? And of course, many of them you have to have some way for cells to talk to one another. And then these, in turn, start regulating the expression or other things of things like morphology, differentiation with different, say, master regulators, as well as the production of new signals that themselves can start giving you cascades of signaling or even feedback. And so what we realized is that we could use, if we could take sort of a simple system that doesn't really self-organize, we could use something like synNotch or different flavors of synNotch to provide novel ways of our own constructed cell-cell signaling. And then we could now, because we can use it to drive whatever we want, we could have it drive different things like cadherins that regulate adhesion, to drive different fluorescent proteins as stand-ins at this point for just the different cell states, and then also even new synNotch ligands that could then propagate in these sorts of cascades. And then we put these into these L929 fibroblasts. These are what Steinberg used for his original cadherin studies, sorting studies. But then we wanted to ask, OK, can we put these in now, have genetic circuits that regulate these, and then put in kind of a simple library of these things, and then ask, could we start getting, do we start seeing self-organization? And maybe at core of one of these questions is, can we make very minimal systems that are only made of these two components, but that regulate one another of cell signaling, cell signaling, and then cell adhesion? Is that sufficient to give you kind of any kind of programmable multicellular structures? And I think the answer is yes, that we were able to get a lot of different kinds of structures relatively easily. Most everything formed really quite interesting, what I thought were pretty interesting things that and so shown here are just a few examples. This is actually a case where we're able to take one cell, put it in. It's actually able to be a bistable cell that can bifurcate into two fates, but then one of them expresses a E-cadherin and sort of sorts into the middle, and it's a dynamically stable structure. This is another one where we're able to take two cells and then put them together. They talk and have a back and forth conversation that eventually leads to the formation of a three-layer structure. One set of cells becomes first a core, a high E-cadherin structure that actually then also signals back out and forms this robust three-layer structure. And then we can also get things like that actually form polarized structures. In this case, we can take the same sort of thing, have these cellular conversations, but they turn on different cadherins that actually sort, and you get you can get these reliable structures that have kind of heads or tails. So I'll just let you look at these movies for a second and then kind of go through a few of the examples just to show how they work. Right, and this is actually one of my favorites. It actually forms these arms almost yeah, we're hoping to actually make those extend and make like a little starfish. Okay, so how do these kinds of things work? Well, just in the simplest case, we can take one cell that has a signal, one cell that has that produces, responds and produces E-cadherin and GFP. And this is a case where these cells don't do anything by themselves, but if we put them together, they start signaling. And again, it takes about 20 hours for these things to happen because transcription isn't super fast. But that these cells will induce the E-cadherin, and then you get, for example, this kind of two-layer structure, nothing too complicated. But then here's a case where we do the same sort of thing. One cell is going to signal, turn on high E-cadherin and GFP. You get a green cell that will form a core here. But then it also, this is actually extracellular GFP, which now actually we have a synNotch in the other one that can receive that signal. So there's this reciprocal signaling that turns on a low level of E-cadherin. And that's what happens. We get this three-layer structure that forms with that intermediate M-cherry structure. And if we put in an inhibitor of Notch, gamma secretase inhibitors, we can kill the formation of that structure. Okay, and this is what I showed before. This is actually where a cell inherently can have two fates of either being a high, either a sender or a receiver, either express high levels of receptor or the synNotch ligand, and switch between this red or green fate, and only one of them also has a high E-cadherin linked to it. And when we do this, and we were actually pleasantly surprised that this worked, we can actually get cells, we can sort out one type, put them together, they come together and differentiate and then start sorting, and they do form a stable inside-outside structure. Okay, and they actually are quite stable in terms of the reproducible percentage of cells that we get. And then this is what I showed in terms of getting asymmetric structures. Here we're driving in this conversation NCAD and PCAD, which will inherently separate, and we can get this sort of these symmetry breaking structures. If we start with low numbers and then grow it in a gradual way, we almost always get this like two-pole structure with a head and a tail. Okay, and then of course, many systems like this can regenerate. We want to see whether we could regenerate these, how stable these were. Cindy Tang is a colleague at Stanford who's developed this microfluidic guillotine. I'll show you that in a second. So we can take these structures, and this is pretty cool. You can run this through that guillotine, get these half structures, and they actually are able to, within a few hours, reassemble, regenerate, and restore that three-layer structure. Okay, so with this minimal toolkit composed only of cell-cell communication coupled to adhesion, we are able to generate very simple multicellular systems that have at least some key things that we think are important in morphology and tissue. We can get they self-organize in a stereotyped sequence of events. They can form discrete spatial compartments and layers. They can drive cell type divergence, where we get an increase in the number of phenotypes over the number of cell genotypes. They can self-repair when damaged and get reproducible structures and ratios of cell types. And we can get symmetric or asymmetric structures. But very importantly, they're programmable in the sense that we can take that limited toolkit of pieces we have, but if we link them together in different kinds of network structures, they yield different structures, different morphological structures. And this just shows a gallery of the kinds of things that we can get from this simple case. And really, we think part of why this works is that as you have cell signaling changing things like morphological outputs like cell sorting, that itself creates more spatial reorganization. And so you iterate, and then new cell-cell positional information. And so you get this sort of iterative increase in distinct positional information by coupling these things together. We think it's interesting in terms of just thinking about kind of what is the minimal toolkit to kickstart the evolution of multicellular systems in animals. Thinking about things like choanoflagellates, I think it's interesting that most individual cells, of course, have ways to sense what's going on in the environment and then to attach to the environment or things in the environment, things like sensing and catching food. And choanoflagellates have things that look like cadherins on the outside, and these are postulated by Nicole King and others to be involved in catching bacterial food originally. But I think once you have those kinds of components, I think it's easy to imagine how these could convert into cells being able to sense each other, as well as cells being able to stick to one another. And I think one of the big points here is that if you actually if you have these and actually start having these cross-regulate one another, you really start moving strongly, have powerful forces that will move towards self-organization. Okay, I want to now talk about really much more kind of open-ended things that we're trying. What are other structural features that we can start to build or explore with these synthetic cell-cell communication and adhesion components? And we are also developing a lot of new kinds of orthogonal adhesion components that have different kinds of flavors of what they do to the cell and tension and et cetera. But I'll tell you about some of the things that is really ongoing, and I'd love to hear your input on this. One is trying to explore this long-standing question of kind of what, the space of morphogen sensing circuits. And then also, can we actually start looking at how changing structures of tissues can actually alter function? And we're starting to play with one system, which is pancreatic islets. So I've talked to you about work using juxtacrine signaling. We can turn GFP into a juxtacrine signal using synNotch. But of course, what we've heard a lot about here is that the spatial range of signals is really important, and we'd like to really have, ideally, things that are much more diffusible, but also kind of intermediate ranges of space, and understanding how interleaving those can give you different kinds of complexity. So one of the things that Satoshi has done is shown that we can actually convert this also into a diffusible morphogen. We have to play with it a little bit, and you can argue that it has its own particular things that are like some biological systems, but we can produce GFP from a secretor cell if we have both receiver cells and what we're calling an anchor cell that provides an antibody here. This is actually, we have two nanobodies to GFP that are compatible. So, but one can actually hold it there like an anchor and then allow cells that have the other receptor to actually read it, okay? And it turns out we can actually make multiple molecules besides GFP into this kind of morphogen. So this just shows that if we put a combination of anchor cells, receiver cells, and secretor cells, then we can turn this M-Cherry response element on. Okay, so we can make short-range contacts, we can have long-range contacts, we have multiple flavors, we can hook them up flexibly to gene expression. This could lead to things like positive or inhibitory feedback. And we think of this as kind of at least the beginnings of a morphogen toolkit where we maybe can start flexibly engineering different kinds of output responses. It's orthogonal, there's no pleiotropy, because the responses to a lot of morphogens are obviously very complex. And then we can also easily express soluble inhibitors. We have a blocking antibody that can be soluble or tethered that actually can prevent the signaling. And then we can, as I said, we now have multiple flavors of these. We haven't done this yet, but we think we can sort of make cascades where you can have transforms of multiple morphogens. So, of course, in this space, and I'm sure everyone talked about this yesterday, but that right now, of course, this is a classic problem. Of how morphogens form in various different contexts. We see this over and over again, and there are a lot of really fascinating things that are emerging in terms of both how the morphogen gradient itself can be reshaped through consumption and transport and other things like that, which we heard about, but then also how cells themselves can actually interpret that information and read in different levels of that. And we know a lot about kind of how that works in example detailed systems. And as I said before, we also have, there's a number of interesting theoretical studies that allow you to actually ask the question more about the whole space, about what are sort of coarse-grained simple circuits that can produce things like, say, a French flag kind of model. But what hopefully we hope to add here is to be able to start building simple genetic circuits that kind of bridge this gap here that allow us to, in a tunable and way we can tune parameters, really look systematically at what are the, how do we catalog and understand the space of possibilities here. And just to show you some of the things we're doing, so this is a case where we're plotting, we're plating, these are again L929 cells, but we're doing it with MDCKs also, a kind of a body patch here. This is all in vitro, and then we have another patch of cells on either side that can be kind of poles that can send signals. And so this is an example where there's diffusible GFP here, and you can see it's hooked up, the response is hooked up to RFP, and we get this nice gradient response. And then in this case, this is a case where we actually hooked up something where the body cells can read GFP, but they also will produce GFP, soluble GFP, which is, again, going to be a signal. And then what you see here is we can get things where Again, it begins in the same sort of way, but then it very rapidly propagates to completely fill up that space. So this at least shows we can start doing some of these things. Here's just a few more examples. This is actually the two things I showed before, more quantitatively, that we can get this spread. We actually can do negative feedback, and actually it leads to a shallow but much more stable gradient. This is a case where we actually put an inhibitor here with the positive feedback, and so this kind of wave goes over there, but it's blocked, and we get much more of a kind of a square wave function. So we are interested in asking, can we start trying to assemble in this sort of generalizable generic system more of a periodic table of possible morphogen patterning circuits, and understanding how you interleave different sorts of things, like what happens at an individual cell versus what happens more globally in the tissue. And then interesting, I also want to point out that we've talked about kind of making an arbitrary molecule into a morphogen. I think it's really interesting to ask whether we can take endogenous signals that might be in a tissue, in a disease or whatever, and actually can we start building systems that actually incorporate that information and turn that into modules, morphogens, to drive or at least start to trigger different aspects of in situ regeneration, which I think is one of the biggest problems with the whole concept of regenerative medicine. Okay, so finally, we've talked a lot about kind of how you form structures, but then I always have to think about, like, well, ultimately those structures are there to perform functions, and if you're talking about a complex organism, that's a pretty hard thing to quantitate. But are there systems where we can actually look at structure and function? And really, we like to think about at least these multicellular tissues. They start talking to each other. Not only do they form structures, but they actually act as multicellular computers that make decisions. Of course, rewiring the brain and being able to put neurons together would be the coolest thing to be able to do. But we realized that even kind of simple things, relatively simple, like islets in the pancreas are important. We all know that beta cells make insulin, but of course there are also these alpha and delta and gamma cells. But in a simple way, glucagon made by these cells actually promote secretion, and then delta cells with somatostatin suppress it. So there's an interesting paracrine kind of architecture that goes on that we believe is used to control things like the homeostatic set point, kind of the bolus of quanta of insulin secretion, et cetera. And interestingly, if you look at different species, the organization of these different cells, and the alpha and beta are the most, and that's what's shown here in red and green, they have really different structures in terms of what's on the inside, whether they're dispersed, kind of what's the distribution of what cells are close to one another. And that is linked to different set points and things like that. You can transplant things into, say, a mouse, and the set point is largely determined by what the origin of species was. Okay, so we just have learned this, very new to this, so I apologize for those of you who know more. But we figured, well, these things kind of look like the structures that we can build. These are some of the structures that we can build looking at higher resolution. And we know we can build things like the diversity that we see in different islets. And so we've started to ask, can we take these, right now they're cell lines of alpha and beta cells, but we've shown that we can actually start putting them together in different inside or out or outside in and different or dispersed structures. And our goal is to now start looking at these both in vitro and then also transplanted into mice to see how the microarchitecture alters the glucose sensing and response. And it's really the first case that we are interested in seeing whether we can really, again, alter the linkages structurally in this multicellular computer, if you will, to get different kinds of behaviors. And that's work that Wesley McKeithen is starting. So just to end, I think there are some really interesting possible applications for synthetic development that, one of the big challenges of the idea of regeneration, of course, regeneration has largely become a field right now with organoids and not as disease models, right, to look at, and less so about like actually repairing or build tissues or whatnot. And I think part of that problem is that, these cells are programmed to have to do the right thing only in the right context of development. And many of those cues are missing. And the question is, can we start understanding what is required for kind of that whole communication and provide cells or provide them with ways of reading endogenous injury signals, et cetera, to replace that and then get back on track on some of the things that we want to do. And I think that this sort of work hopefully will lead to some tools that facilitate that. And then also, especially as we think about like the pancreas, are there synthetic organs or tissues? Might it be better to have the flexibility to change what the system does that could be better. And as an example, of course, even in diabetes, we spent a long time trying to clone human insulin and make recombinant insulin, but then those have been completely replaced by engineered insulins that have either long or short half-lives, and those are much, much better for therapeutic use. So if we have that ability, we can maybe address things in that way. Overall, we think that synthetic development can help us to try to test and validate fundamental models of development and better understand the design space of how we write these sorts of morphological codes. And that also will inherently, I think, give us much better capabilities in asking how we can do things in terms of tissue regeneration, repair, and replacement. And so let me thank the people who did the work. Most of this was Satoshi. The synNotch system was developed by Leonardo and Cole, and Wesley is doing the islet work. I talked about Cindy Tang and the microfluidic guillotine. And with that, I'm happy to take any questions.
Todd, down in front first, and then we'll circle around for the rest of the questions.
Thank you, Wendell. So I have two questions. First is have you tried to do the combination with the programming of the cellular structures with stem cells to see if it stabilizes differentiation, accelerates differentiation? Anything with starting to do these types of organizational things in stem cells? That's the first question.
Yeah,
Second question.
Okay, go ahead, finish.
Okay, second question is kind of similar, which is there's an opportunity here, I think, with respect to the programming of these cellular forms in the context of either natural or possibly synthetic scaffolds.
Scaffolds meaning materials?
Yeah,
Okay,
yeah, materials, so that you can make these more, higher functioning and more complex tissues.
Right. Yeah. Yes. So I would say first we have put the synNotch system into ES cells, and we can, for example, drive certain factors that will lead to sort of semi-differentiation towards different lineages. So they do work in there, and we certainly could start trying to impose new programs on kind of intrinsic developmental networks inside the stem cells. Something that we're actually a little bit more interested in is the idea of building kind of synthetic organizers or niches, so that we actually don't necessarily touch the ES cells themselves, but kind of build whatever might be the trifectoderm or other niches, depending on what stage. There's always kind of a chicken and egg thing where there's a context that helps to steer them. But I think that is an interesting idea to us of trying to actually take a native system that has potential paths, but can we build a sort of a partner system that will steer it in certain ways. And then the materials and the scaffolds, yes, we definitely can attach molecules to a scaffold and then have them be recognized. So that is, we can incorporate that element.
Rick in back, and then we'll.
Hi, I was really intrigued by your notion of trying to kickstart regeneration.
Yes.
Can you put a timer in there so that you would kickstart regeneration and then generate apoptosis?
Yeah. Yeah, so we certainly have been playing with both of those elements. I mean, our lead applications are mostly in immune cells, where we're trying to do things like that, and we are trying to control. We are able to control like expansion and contraction of populations of T cells through controlling things like apoptosis. So we've done a little bit on that, and we are very interested in timers. I'd say that and how to do that robustly, because I think, to me, having control over time and space in terms of length of action, if we have those, we're in pretty good shape.
Person in the black shirt, then we'll get this side of the room.
What do you think is the functional significance of all these different spatial distributions of these pancreatic cell types across different species? Like, why are they so different?
Well, I mean, they do have different set points, and I think they have different. I mean, there's a lot of really interesting things. I mean, they're excitable systems, right? So they lead, and gap junctions lead to sort of spreading. So how they're clustered may control kind of what the bolus of one kind of subpart secretes, insulin, and of course the neighboring cells will set up the feedback that controls the set point. So I think no one really has been able to study that, and that's kind of the point.
Question over here. Yes.
And I'd love to find out other kinds of simple scale things that are kind of, again, multicellular computers where we have some evidence that this architecture is controlling a higher order function. Yeah.
Yeah, I like the fact that you actually included engineering.
Yeah.
But I was intrigued that you took the evolution out, and you said that your system must be clean, and then obviously you have a lot of mileages.
Right. And of course, that's not completely true, but go ahead.
Yeah. You have already started doing evolution. So what does it take to actually create an environment where you do combinatorial mutagenesis, where your cells actually evolve under a given set of experimental conditions?
Yeah.
Obviously, this would be very useful for cancer therapy.
Yeah.
You may then be able to reverse the evolution and then cure.
Right, yeah. So I think that's a great question. I mean, the problem with evolution, of course, is it's opportunistic, and it doesn't know where it's going, but it finds opportunities that are advantageous. So when you think about things like morphology, that's a really challenging beast to come up with some sort of a selection and whatnot. I mean, we are able to take things like this and say in the context of immune cells that are fighting cancer, if we look at something like what drives proliferation of the T cells, that we can start looking at enrichment of different combinatorial libraries of circuits. In the case, we can also, sort for different sort of lineages that come out and ask, what are the circuits that favor that? So I think that's one of the big challenges. Most in vitro evolution, if we're trying to do that kind of thing, is selecting for survival or a color or something like that. And I mean, there's just higher order function that we need to be able to look at.
One last question. The microphone will come to you in a second. In the hat. There you go.
Are you saying then that you have created a synthetic living cell that moves on its own volition?
Well, I mean, that's a complicated question. We're working with cells that exist that lack certain higher order organizational properties. I mean, there's a whole other branch of synthetic biology that is trying to look at, like, can you make a minimal cell? So that you can ask the question at many different levels. I'd say that we are working with cells that have their standard kind of central dogma operating system and other things like that. So we're definitely, riding almost entirely on the backs of evolved systems, but trying, I think, to ask where's the point where we can put regulatory systems that control their organization in a multicellular way. We have not really used a lot of, say, migration or even intrinsic cell polarity, like what Verona talked about, that are obviously important. Although I'd say that when we do start doing things with like ES cells and put little organizers there, they induce things, and definitely cells. A big part of it is that certain cells start moving, right? So I think there's a vast space of possibilities and different levels to frame the question in terms of how much you're looking for. I think what we're interested in is more kind of trying to impose regulation at a certain scale within a broad multi-scale system and ask kind of how that changes it.
Okay. Thank you so much.
Okay.